Okay, well, I guess we'll go ahead and get going. It's my pleasure to introduce our speaker for today, Loren Rieseberg. Loren got his MS in botany from the University of Tennessee, went on to get his PhD also in botany from Washington State University. His first faculty appointment was at the Claremont Graduate School, and he's currently the Distinguished Professor in the Department of Biology at Indiana University. And he's also the Canada Research Chair of Plant Evolutionary Genomics at the University of British Columbia. And Loren has done some amazing research. We're going to hear a lot about it today. His primary foci of research have been speciation, also domestication and genomics, obviously. He's used Helianthus basically as a model system. And in that, he's found that speciation is largely driven by hybridization, ecological divergence, and also chromosomal rearrangements. And if you look at his CV or you browse around the internet a little bit, you're just kind of amazed by the amount of accolades that he's accumulated over his career. He's a certified genius. He's received the MacArthur Fellowship, also known as the Genius Grant. And just last week, he's joined this kind of rarefied group of individuals, including R.A. Fisher, J.B.S. Haldane, and Ernst Mayr, in winning the Darwin Wallace Medal. So it's from the Linnean Society of London. So that's basically acknowledging his contributions to plant evolutionary biology. So please join me in welcoming Loren Rieseberg.
I want to thank Matt for a very nice introduction, which I spoiled with this mic thing. And it's really a pleasure to be here, although Mike Otrelli told me today that, just before the seminar, that I should not use the term Bateson-Dobzhansky-Muller incompatibility because Bateson, so I apologize for using that term yesterday. Michael is actually right. It should be probably just DM incompatibility, so if I make that mistake, let me know. I also want to mention that I'm very pleased to see that one of my first grad students, Peter Morell's here today, so that's great. And finally, I've had a wonderful time here. Yesterday, I was given a UC Davis hat and a DNA Helix lamp, which was very nice, so there. So it's been a real pleasure. It's remarkable to me how well UC Davis has stockpiled talent in ecology and evolutionary biology. You should share a little bit. It's like, you know, you could take the talent at Davis and scatter it over the middle America and sort of, yeah, anyways. It is remarkable to me, and there's just a huge amount of strength, and I've had fun talking to everybody. So today I'm going to be talking about the genomics of plant speciation. Just sort of, it's, as I've told people during my visit, the technology's changed so much during my career in the last 20 or so years that I feel like I could do everything, my entire career's worth of work in 2 years and do it a whole lot better now. So it's really intimidating, and it's a lot of fun though, because now we can really address some of the questions that I'm interested in. Over the next several years, I hope to do so. Although everyone is getting there first through, you know, sunflowers have very large genomes, so the Drosophila folks and the Arabidopsis folks, and they're all ahead of me, and the maize folks are several years ahead, and it's kind of discouraging. With that said, I'm going to introduce speciation. I think that you all obviously know what speciation is. In my view, at least what I'll be talking about is speciation is simply a process characterized by the evolution of barriers to genetic exchange between previously interbreeding populations. And basically, this reduced gene flow facilitates the accumulation of genetic differences that can result through stochastic processes such as genetic drift or through selection. And mainly, I think, through selection. And I think it's important to remember that this is kind of a feedback loop where you have reduced gene flow, which facilitates increased divergence, which again is going to reduce gene flow further, and so on and so forth until speciation is complete. And in the past decade or so, with these revolutionary advances in high-throughput sequencing and computational biology, it's now possible to ask questions we really didn't think about several decades ago. And one of the things we can ask is sort of just how do genomes diverge during the process of speciation? And one of the first models that was put forward was by Cheng-Yi Wu as kind of a conceptual model, but it's still, I think, useful to talking about what we might expect. And this is a model for speciation with gene flow, actually. And Cheng-Yi suggested that he sort of divided speciation into several different stages. And a lot of other people have done the same thing. Some people have 3 stages. Some people have 5 stages. Some as high as 10 stages. But Cheng-Yi Wu had, I think, 4 stages. And he suggested that initially what you have is you just have the sort of local adaptation. You have differential adaptation where you have a handful of loci that are responsive, that are under selection, under divergent natural selection. And then there's no gene flow at these loci and very closely linked to them. And then the rest of the genome is connected by gene flow. And in stage 2, according to Cheng-Yi, you get some populations with some reproductive isolation. At this point, the populations may fuse or diverge. And his idea that these regions that are protected from gene flow begin to expand, and the regions that are where gene flow is allowed, or where gene flow continues, becomes more smaller, and the amount of gene flow, of course, is reduced. And then in stage 3, you can see that now a major fraction of the genome is now sealed from interspecific gene flow. And he calls these good species. There is some hybridization, but basically the populations are now beyond the point, or the species are beyond the point of fusion. And then finally, in stage 4, he would call these species with complete reproductive isolation. And although this wasn't the point the main point of Cheng-Yi's sort of paper, is that this idea that these regions of the genome regions of divergence expand during speciation sort of caught on. And there was an important paper here at UC Davis, actually, by Tom Turner and Matt Hahn and Sergei Knudsen. I think it was in 2005 in Mosquito. And they basically called these things speciation islands. And this term really caught on. People liked that terminology. In fact, in 2012, there was a whole issue on sort of these islands of divergence in the Philosophical Transactions of the Royal Society. It came out in February. And there, the language really ran amok. There were not only speciation islands, but there were speciation archipelagos. There were speciation continents. I mean, it's really great language, but it's kind of, it's interesting. The thing of it is that we really don't know if this is what happens. In fact, a lot of people are skeptical that this is really what goes on. Another model might be that, what happened here? Oh yeah, this is right, sorry. I didn't sleep so well last night. So another model might be that instead of getting these speciation islands, what happens is you simply have more loci that are divergent due to differential adaptation, and that you just get more and more and more until the genome is sealed off from gene flow entirely. And we really don't know, for example, which of those models occur, how big these islands are, if they exist, and so forth. And nor do we even know whether gene flow is responsible for, interspecific gene flow is responsible for the formation of islands or not, if they exist. So this is basically what I'm going to be talking about today. I'm first going to ask, and we're going to focus on sunflowers, and it could be that things are going to be very different in different organismal groups. But I'm going to ask whether or not these speciation islands really exist. If so, how large are they? There have been reports in the literature, often from low-resolution scans, of these islands being as large as 50 centimorgans, or half a chromosome. Whereas a lot of the high-resolution scans, they really don't find those kinds of large regions. And how does interpopulational gene flow affect patterns of genomic divergence? In other words, if we see islands, are they only found in species with gene flow, or are they found in everything? And what are the causes of these genomic islands of differentiation if they exist? And then the second part of the talk, I'm going to focus on a topic that a lot of people become quite interested in. And there's a lot of interest in parallel adaptation and finding genes that have, or SNPs that have over and over again been involved in adaptation. And so I'm going to ask is how repeatable are patterns of genomic divergence across independent pairs of species? And then I'll talk a little bit about what the causes are of the patterns of parallel patterns of divergence. And I'll suggest that it's not all just parallel natural selection. Okay. So a little bit of theory just to get us started. So we know that now that the genetic differences between closely related species mainly arise via positive selection. And this is something called adaptive hitchhiking, right? I think we all know this. And the size of the differentiated region, right, when you undergo a selective sweep or adaptive hitchhiking, tends to be quite small. And it mainly depends on the selective advantage of the favorable mutation relative to the recombination rate. So if you have a mutation that has is very strongly selected, and there's low recombination, you'll have a much bigger region. But even so, the size of the differentiated region from adaptive hitchhiking is expected to be very small. However, there's another way that this might happen. These differentiated regions may expand due to reduced interpopulational gene flow near loci under divergent selection. And this is called divergence hitchhiking. And the basic idea here is that if you have populations exchanging genes, and alleles come from, and maladaptive alleles are constantly entering the population, and as selection eliminates them, it actually replaces them with the local allele. And what happens then is linked neutral sites to that local, or even adaptive sites to that locus, actually will increase in frequency, and so you can expand these regions of divergence. And it's through these models that folks suggest that maybe you can even get regions of divergence extending, many centimorgans. So there's been some models, some theoretical work on this, and it actually shown that divergence hitchhiking does occur under some conditions. There's also some older models as well. But in other cases, what you get instead is that interpopulational gene flow actually reduces the hitchhiked regions. In other words, it erodes neutral variation, the neutral link sites. And so it's almost more of an empirical question. What do we actually see in nature? An important point, though, is that divergence hitchhiking requires interpopulational gene flow. In other words, you have to have this interpopulational or interspecific gene flow, whereas adaptive hitchhiking does not. And it's interesting that many of the papers that have looked at these genomic islands, these islands of speciation, have just assumed that the patterns of genomic heterogeneity across the genome are simply a result of gene flow, when they could just be due to selective sweeps and adaptive hitchhiking. You really need to compare what happens in sympatric and allopatric populations to actually know what is the consequence of interspecific gene flow. So this is the study system. As Matt mentioned, this is we do most of our work in sunflowers. There are some good features and bad features about sunflowers, like with every other organism. It's a pretty species. That's good. It's native to North America. There are about 17 chromosomes, although it's genetically diploid. There have been at least 3 paleopolyploidy events in the last 50 to 60 million years. So it has a highly redundant genome, which is not good. And in fact, if you go back to the origin of angiosperms, you're really talking, we'd probably make it, 16 or 18 ploid, okay? So there's lots of redundancy, lots of paleopolyploids in its history. The most important ones are those that occurred in the Compositae family. It's a self-incompatible outcrosser, so in that way it's kind of like an animal, which is good. It fits population genetic models, not like a selfer. It's adapted to different habitats, and so clearly ecological adaptation to local environments is one of the first changes that sort of initiates the speciation process. Fairly recent diversification, and also unlike a lot of really closely related species, sunflowers have both strong prezygotic and postzygotic barriers. Basically, hybrids between the species typically have less than 10% fertility, and often even less than 1%, but you still get gene flow. Hybridization is frequent, and there's a very high rate of chromosomal evolution. I'm not going to be talking about the chromosomal rearrangements today because with this new high-resolution data, we're trying to get things figured out. It's more complicated than I thought in the past, is really what it amounts to. And if you have questions about it, I can tell you where we are, but we still have a ways to go to figure everything out. So these are the 4 species we're going to be talking about today. There are 2 sets of sister species, Helianthus debilus and Helianthus pediolaris. And debilus and pediolaris actually have fairly similar habitats, except that debilus occurs in Texas and pediolaris occurs in the Great Plains. And then the other sister taxa are Helianthus argophyllus. This is a sister leaf sunflower. It occurs in Texas. It occurs in sandy soils, but they're much wetter in the spring than those found in Daedalus. And then Annuus is this Great Plains species that you all are familiar with. It's the progenitor of the domesticated sunflower. And it tends to prefer sort of mesic clay-based soils that often are quite wet in the spring. In other words, they'll be in a ditch or something or in a cultivated field. And then they dry out later in the summer. It also occurs in natural habitats, often naturally disturbed habitats. And this gives you sort of a timeline. These are fairly recently diverged species. Daedalus and Pediolaris are probably somewhere between 500,000 and 1 million years diverged. Annuus and Argophyllus are a little bit over 1 million. And then the whole group is about 2 million years in divergence time. That assumes that our mutation rate estimates are somewhat accurate. And that the generation times of an annual sunflower really is about 1 generation per year. They have a seed bank. It could be closer to 2 generations per year. So take those dates with a grain of salt. Just use them as a relative date. OK, so this is the native range of the taxa to be studied. As you can see, Annuus and Pediolaris, here and here, the gray and the blue, in different clades, you can see they have this broadly sympatric geographic distribution. And so whenever these species come into contact, they hybridize. And there are literally millions of small hybrid swarms or hybrid zones. The hybrid zones, because there's a very strong sterility barrier, F1 hybrids actually have about 1% seed set. The hybrid zones are extremely narrow, like 10 meters. They really are these just sharp transitions between the two habitats, between the species. So Argophyllus, the two pairs of sister taxa are allopatric. And then Helianthus annuus, in bright blue here, is parapatric with Helianthus debilus. And debilus and Argophyllus, although they are parapatric here, they actually do not exchange genes. There's no hybridization between them. So we'll treat them as allopatric for this talk. OK, so the basic methods, we did this before. I didn't mention one of the big disadvantages of sunflowers. They have a very large genome, 3.5 gigabases. And we don't have it quite put together yet. It's mostly made up of LTR retrotransposons, and most of them are less than 1 million years of divergence, and so it's turning out to be a real challenge for us. Big disadvantage, and so at this time it was much cheaper to be doing transcriptome sequencing, so we've done a lot of transcriptome sequencing, both with the Illumina and 454 sequencing platforms. Initially, we did this mostly with normalized libraries because it was cheaper. Sequencing costs were still quite large. And then as things got less expensive, we just use RNA-seq or conventional libraries on the Illumina machine. And in some ways, there are both advantages and disadvantages to this. Having the RNA-seq data is really nice because we can look at expression versus sequence divergence, which can be useful for interpreting these patterns. And this just shows the number of genotypes that we're comparing here. So in terms of the data analysis, because our reference genome is not yet complete, and I keep on saying it's going to be about 6 months. It's going to be about 6 months. But anyway, so we've been using a genetic map created by one of my, he's now a grad student. He was a bioinformatician initially. And he kindly put the genetic map in the shape of a sunflower head, just so if you don't know what that is, that's a sunflower head. And it's kind of a cool map. It's based on just sequencing recombinant inbred lines to 1x depth with Illumina. So you basically have a map of about 20% of the sunflower genome is placed on the map. And so what each of these concentric rings is, this is all the recombinations in a given recombinant inbred line, with the black bars coming from one parent and the white bars coming from the other parent. So you can actually look at the map and see every recombination event. And then each of these bars here is actually each chromosome. There are 17 of them. These little blue lines there are each of the bins, or each of the recombination breakpoints. And then the yellow rays indicate the number of base pairs that map to a particular centimorgan window. And so when you see a lot of them mapping to a particular window, this is what we think is where the centromeres are. But we don't know that for sure. We haven't mapped those. OK, so the data analysis, we've used FST, which is a measure of genetic differentiation. And essentially, we've calculated for all 6 interspecific comparisons. we're looking at between about 190 and 270,000 SNPs per comparison. And then we've conducted 1 centimorgan sliding window analyses to identify genomic regions with elevated FST, using sort of permutation tests to find significant regions. And we've also employed spatial autocorrelation to examine the clustering of outlier loci. In this case, we just decided to use an arbitrary 3% of the most divergent SNPs. And that's usually, for the more divergent species, that's all of the fixed differences. And the idea was to try to be fair in terms of finding islands of differentiation. We get almost exactly the same results if we use 1% of the most divergent SNPs or 5%. But you can view that most of these probably, well, we'll see later that most of these were driven to fixation by selection. And then we also calculated alpha. And in this case, alpha simply refers to the proportion of base substitutions that were fixed by natural selection. We did this for the outlier loci. And we used the basic equation. This is simply a modification of the McDonald-Kreitman test, where you're really looking for an excess of non-synonymous to synonymous substitutions. OK, so this gives you a basic sort of overview of the data. And on the y-axis, we have the timing of divergence. On the x-axis, we have gene flow. So you can see that so we have the more ancient divergences up here. And then if you look at gene flow, we have allopatry to sympatry. So we have very high levels of gene flow to very low levels of gene flow. And you can see for allopatric species of recent divergence, you have an FST of about 0.237. And if you look at the FST of more divergent species that also are allopatric, you get about 0.46. If you look at species with some gene flow, Anuus and Pedularis, they're highly divergent, and they're completely sympatric. We get a lower FST value, not surprisingly, and an intermediate FST value for the parapatric species pair. So the overall average FSTs make sense in terms of what we know about the divergence times and the amount of gene flow between the species. And this gives you an idea of what the distribution of FST looks like. So on the y-axis, we just have the frequency of SNPs, and on the x-axis, we have FST, the major differentiation. And you can see in the sympatric pair, we have a lot more zero FST values, or very close to zero, whereas we have the whole distribution is shifted to the right for the allopatric pair. And again, that's about what we expect for sympatric and allopatric species of the same divergence time. OK, this slide has way too much information on it, just to show you it exists. So these are just looking at FST values across every single linkage group, all 17, across all of these 6 comparisons. We have the sympatric, parapatric, allopatric species pairs. And these black areas simply indicates regions or islands of differentiation. That is, these are regions where we have higher than expected. These are sliding windows where we have higher than expected FSTs. And the important thing to note is that we see many of these regardless of whether they're in sympatry or allopatry. And so we have many islands of divergence. But it's important to note that these islands only have a small part of the action. They only include 7 to 10% of all divergent loci. So most of the action is occurring outside of these islands of differentiation. So a couple conclusions that we can make here by comparing among taxa. First of all, we find that the genomic islands are really quite small. We don't have any. We have a few islands that extend. There's some up around 2.5 centimorgans, but most of them are very, very small, with the average in all taxa being about, all pairs being about less than a centimorgan, in fact, about 1/3 of a centimorgan. There's also no difference in island size between sympatric and allopatric populations. So we're not getting these extended islands of differentiation in sympatry that has been predicted by some people under the model of divergence hitchhiking. It just doesn't happen in sunflowers. And also, there's no difference in the number of islands that we're seeing in the sympatric and allopatric populations. They're all about the same. The one thing we did find, though, is that when we looked at the clustering of outlier loci, we found that there was a difference between sympatry and allopatry. These outlier loci are much more clustered in sympatry than they are, or parapatry than they are in allopatry. So there is a difference, and this is actually predicted by the gene flow. The problem is that it only affects very closely linked loci that are really tightly linked. We don't see it across broader centimorgan distances. So what's happening? Well, we find that if we look at alpha, we find it in the sympatric and parapatric pair, that there's much greater proportion of outlier loci are adaptive. In other words, alpha is much higher in the comparisons of our sympatric and parapatric taxa than they are of our allopatric taxa. And some of this effect is due to effective population size, but not all of it, because we have both. We have species with large effective population sizes in the allopatric comparisons as well. So let's try to reconcile this a little bit with theory. And I'll do my best I can. Some of you might be able to do this better. You can tell me afterwards. So the fact that we have very small islands of differentiation and that they do not differ in number or size between sympatric and allopatric pairs of taxa, in my view, this supports adaptive hitchhiking as the main reason that we're actually seeing these islands. However, we do find greater clustering of outlier loci in the sympatric taxa. And so this could be used to support a role for divergence hitchhiking as well. However, I don't think so. And the reason is that we actually find that there's alpha is higher in the sympatric and parapatric taxa, which suggests instead that what we're seeing is the clustering is the result of the erosion of neutral and weakly selected differences due to interspecific gene flow. So I don't think we have any evidence of divergence hitchhiking, actually. But we might. Actually, I wanted to say a little bit more. So, well, maybe I'll talk about that at the end. So the question is, well, I think there's sort of a general question here I haven't addressed is then, why should, if all of this, if these islands are due to adaptive hitchhiking, the question is, why is it clustered? Why do we see any clustering at all? And so, because presumably they should just sort of be randomly distributed across the genome. In fact, there should be interference between selective sweeps if they're too close together. So what's going on? And in my view, the reason, it could be that some of this is due to small inversions that we just don't know about. But I think probably most of it is due to the fact that when you have gene flow between populations in different habitats, there's weak selection for linkage. And this weak selection for linkage has been going on ever since the Permian. So we've had this weak selection for linkage, and I think what it is that locally adapted alleles tend to be linked, at least at the very, very local spatial scale. And I might be wrong about that, but that's how I interpret the fact that we have islands of differentiation. It's not, they're due to adaptive hitchhiking, and they're due to the fact that locally adapted genes or genes that are under selection often functionally related genes tend to be linked. Okay, now I want to talk a little bit about the repeatability of speciation. And so what I'm going to be talking about here is how repeatable are patterns of genomic divergence across independent pairs of species. And then I'm going to try to tease apart the causes of parallel patterns of genomic change and do this second part fairly unsuccessfully. Okay, so to put the big picture again, remember there's a lot of arguments about the repeatability of evolution. Stephen Jay Gould argued that, at least at the macroevolutionary scale, that rewinding the tape of life would produce a very different array of end products. This was in his book, The Wonderful Life, about the Burgess Shale. But of course, others have reported, particularly at the microevolutionary scale, have reported that the repeatability, that the evolution of some traits and species can be surprisingly repeatable. And I think we all know examples of this. In BC, we always see these stickleback fishes that have undergone the same parallel speciation events over and over again. The examples of mimicry are quite remarkable. Here you have different species look alike, but within species you have strikingly different forms. And we actually know almost very, very little about the repeatability of evolution at the genomic scale, but this is the scale that's going to be very easy to test. because we can do this now. And I think we're going to find that evolution is remarkably repeatable. And that's what I'll be showing today. So there's a little bit of theory, and I hate to say this because I'm using one of Alan Orr's papers, and Alan is a hero of mine, but I've heard that Graham Coop has a recent paper published last year, the year before, that actually is sort of a more sophisticated version, sort of an update of this, that adds more different factors. And I wasn't aware of Graham's paper. And apparently, though, he's talked about it. He's been working on this with one of my postdocs, and I didn't know about it. So I'm kind of like, I should have had Graham's picture up here instead, and his, especially since I'm at Davis. But anyways, Alan did a very nice paper on the likelihood of parallel evolution or parallel adaptation. And he was looking at the likelihood that individual nucleotide substitutions would happen in parallel in different populations under the same selective pressures. And what he showed was a very, very nice result, that the frequency of parallel evolution, P, is simply is very tightly correlated with the number of beneficial mutations available. And so all this means is that if you have very few beneficial mutations possible or available, and that would mean that your target for mutation is very small. So if you have a trait with very simple genetic control, or an organism with a very small genome, like a virus or a bacteria, you might expect much higher levels of parallel evolution than an organism with a big genome, a highly redundant genome. And so plants would be sort of the worst possible scenario, because typically you have these genetically redundant genomes, lots of different ways for the same thing to happen. And, but there are other things that play a role as well. For example, the role of standing variation and migration. And apparently Graham's paper takes some of these things into account. So I'm kind of embarrassed that I wasn't aware of it until this visit. So what I'm going to do here is take the same data set, except that I'm going to be looking at patterns of divergence between this sister pair of taxa and this sister pair of taxa. And these are not what we call parallel speciation events at all, okay? But there is actually some parallel natural selection in all likelihood, because this species is found in Texas, this one in the Great Plains, this one in Texas, this one in the Great Plains. So there must be some presumably parallel natural selection going on. And so this is just a very crude analysis of repeatability. And what we're looking here is we're looking at mean FST across the genome. And here's chromosome 1. And so Annuus and Argyrophilus, these are the more divergent species pair, right? And they're in blue. And Pedularis and Debolus is the less divergent species pair in red. And here we have FST, the measure divergence. Here is the chromosome. And what you can see is that the patterns are strikingly similar. Obviously, the less divergent one is not quite as divergent, but you see the same peaks over and over and over again across the chromosome. And if you look at chromosome 3, and the R-square is actually 0.62, so a remarkable level of similarity. If you look at mean FST along chromosome 3, it's quite different. Here we do get a significant result, but the R-square is only 0.1. There's not near as much similarity across the chromosome. So this just shows across linkage groups. I showed you the two most extreme values. So here you have linkage group 1, R-square of 0.6, and then linkage group 3, R-square of 0.1. But you can see there's a great diversity across the different chromosomes and the amount of similarity in terms of FST. And the overall average is about 0.3 or something like that in terms of the R-square. So if we look at the repeatability of divergence, and this is, we're just looking at FST, not the outlier loci, but all loci, all SNPs. And what you see at the sliding window, we have an R-square of 0.33 across the genome. In terms of genes, we have an R-square of 0.14. And then in terms of SNPs, an R-square of 0.01. So all these are highly, highly significant for sliding windows in genes. We have very high levels of repeatability for both, and essentially no repeatability at the level of a SNP. So now we can move over onto outlier loci. These are the ones that presumably have been established due to adaptation. And if we look at the outlier windows, we find that about 4 times as many are shared as expected by chance, 13 to 3. Those are indicated in these sort of orange, not sure if it's orange or pink, windows in common are shown here. In terms of outlier genes, we have about 3 times as many as expected by chance, 220 compared to 88. And in terms of outlier SNPs, it's only about 50% more than expected by chance, 88 compared to 55. So why are these patterns of genomic divergence repeatable? And this was a puzzle that still remains a puzzle to me. And I'm going to go through some possible explanations. In model species, such as Drosophila and Arabidopsis and yeast and a few other species, protein divergence measured in terms of DNDS or KAKS has been shown to be positively correlated with gene expression specificity and negatively correlated with expression level. What that means is genes that have very specific, they're expressed in a single tissue, and these tend to have low overall expression, diverge faster than genes that are broadly expressed across all tissues. And they tend to also have very high levels of expression. So there's strong correlation between specificity and expression level. And so we actually do see this same pattern where we do see a weak correlation between protein divergence and gene expression. And you can see this over here, gene expression specificity. And we do have a weak correlation with DNDS, a positive one, and then we get a negative correlation with expression level, just like what people have found in Arabidopsis, Drosophila, and yeast. However, we get no correlation with FST. And it's FST that we're looking at. And I don't know why that is. Someone might be able to. So I'm going to throw this explanation off the table. So one other possibility, at least for the outlier loci, is that what we're seeing is the consequence of parallel natural selection. And although I've emphasized before, these are not parallel speciation events, we do have this temperature, moisture, and photoperiod gradient that's actually shared between these 2 species pairs. So presumably, we are seeing some effects of parallel natural selection. And I think that this parallel natural selection may explain the excess number of SNPs that have been fixed in parallel between the 2 species. So some other possibilities that I've been thinking about and talking to people about, and this is the one that I've come to sort of favor as one. I think this has a pluralistic explanation, okay? I don't think any one explanation explains all this. But one possibility, and I think that this has to be the case, is that some genes simply matter more than others. Are simply more important in evolution than others. And that this importance is then correlated across species. Also, we know that independently divergent lineages likely share heterogeneity of mutation rate, right? And so genes or genomic regions with higher mutation rates are going to diverge faster as long as the lineages are allopatric. And these are allopatric lineages. So we don't have gene flow between them, so this should hold. However, the problem with this hypothesis, I think it must be, but DS, the synonymous substitution rate, is not correlated with FST in our system. So I don't have any evidence for it, even though I think it must be partly responsible. Also, we know that these independently lineages are likely to share heterogeneity and recombination rates. And we know that deleterious mutations are culled less efficiently from regions of low recombination. And so this should presumably explain part of the pattern we see. However, our problem here is, first of all, we don't have absolute recombination distances because we don't have physical distances for all of our genes. But we do know that in general, gene density is often fairly strongly correlated with recombination rates, and we see no correlation between FST and gene density. Another possibility, and I think again this must be, I think all of these must be, is that genes vary in selective constraint, and such constraints are correlated across independently diverging lineages. However, probably the most important selective constraints is expression level and breadth, and we see no correlation with expression level or breadth. So this is a very unsatisfying seminar, I suppose. I do want to say, though, that the genes that have, the SNPs and genes that are shown to be under positive selection in both lineages turn out to be just a treasure trove of of cool genes that people have been trying to clone in sunflowers and have failed so far. For example, one of the genes a lot of people have been going after is the restorer of cytoplasmic male sterility. And we know exactly where it maps. And people have been getting closer and closer to cloning it. Well, we happen to show up in both of our, one gene that showed up in both of as under positive selection in both lineages was a PPR protein, which these PPR proteins are what's responsible for the restoration of cytoplasmic male sterility in most plants, and it maps right to the restorer locus. So I'm guessing that we have it. We can't prove it, but it's a pretty good bet. Another gene that people have been going after for a long time in sunflowers and the composite is the S locus, and is involved in self-incompatibility. Well, we happen to have an S protein receptor kinase that maps exactly to the S locus. And we haven't proven functionally that these actually are the genes, but they're pretty good correlations, and they're under positive selection during the divergence of both lineages, which is exactly what we'd expect for both examples. And there are other examples like that. There's a flowering time gene I've been trying to get at, and it shows up on our screen. There's also, well, I'm going to stop that because you can make these stories forever, and I don't like doing that. But let's just say that even though we're not thrilled about not being able to explain this pattern, we still have some tests to do. We are thrilled with some of the genes that have come out of those screens. So, to conclude, in terms of speciation islands, we find that they do exist, which is cool. But they tend to be small, at least in sunflowers, and they do not increase in size and number and sympatry. These islands of differentiation contain an excess of non-synonymous substitutions, and so both of these factors imply that adaptive hitchhiking is the main proximate cause of the islands of differentiation. And since we know that the clustering of loci favored in adaptation is favored by the evolution of heterogeneous environments, by evolution in heterogeneous environments, I think that this is probably the ultimate cause of these speciation islands. It's also one of the reasons that, for example, we expect to see lots of inversions involved in local adaptation. Although I've been talking to Jeff Ross-Habera's group about all of their inversions they're finding in maize. I think we're going to find these in sunflowers. We already know about some interspecific inversions, but I'm hoping to find some of those polymorphic within species. And so I think that this is why we're actually seeing the speciation islands. We can't rule out the possibility of divergence hitchhiking because we see these greater clustering of outlier loci and sympatry. But this, we think, because we see a larger alpha or a larger fraction of non-synonymous substitutions in the sympatric taxa, outlier loci, we think that this is actually, rather than divergence hitchhiking, it's simply the erosion of neutral or weakly selected differences due to interspecific gene flow. So why isn't divergence hitchhiking working in sunflowers? I think I know why. I think it actually does work, but not in the way, but because of the way we've been doing our sampling, it doesn't work. And the reason is, if you look at all the models that have been done so far of divergence hitchhiking, they have panmictic populations that are exchanging genes. So these independent lineages, but they're panmictic within the lineage, and they exchange genes at some low rate. Sunflowers have subdivided populations. So we might be getting, there are these large geographic distributions. So we might be getting divergence hitchhiking within an individual hybrid zone. But because we're sampling across the entire geographic range of the species, it's going to be a different set of loci, or a different set of neutral variants that rise to high frequency within each local hybrid zone. And so I think that's why we don't see it at all. It might be that divergence hitchhiking works, but go to a lake, a small lake, then you might see it. Or go to something that flies around a few bushes or something where you have panmixes within those populations that are exchanging genes. Then you might see divergence hitchhiking, but not in species that have these large geographic substructured ranges. Okay, in terms of the repeatability of speciation, we found that genomic divergence is surprisingly repeatable at the level of genomic regions, individual genes, but somewhat less repeatable at individual SNPs. And I think that this should be a cautionary note for the use of sort of these repeatability patterns for evidence of parallel adaptation. I'm not sure. I think there are a lot of reasons we get repeatability that might not be due to parallel natural selection. I do think if you find the same SNP being fixed by natural selection over and over again, I'm perfectly fine with that being as evidence of parallel natural selection, but not for individual genes or genomic regions. So why? I think that it's probably caused by parallel natural selection to some extent, the fact that some genes matter more than others, as well as shared heterogeneity and mutation rate, recombination rate, and selective constraints. But as I pointed out earlier, we actually have no evidence for any of these so far in sunflowers. And I wish we did, but we don't. So just to acknowledge, Sebastian Renaud is one of my postdocs who did most of the analyses here. Nolan Kane and Chris Grass are also, Nolan's a postdoc, Chris is a grad student. They did a lot of the SNP calling and original sort of bioinformatics. Quite a number of folks helped gather data, did the transcriptome sequencing. I've had long discussions about these data with Anil Agrawal and Mike Whitlock, trying to keep from making the wrong conclusions. I probably still have, so they shouldn't be blamed. And Rose, Andrew, and Sam Yeaman are postdocs that also have had good discussions with. So thank you very much.
We have time for a few questions.
So have you looked at the genealogies of these shared window of divergence? Can you tell whether they're due to recurrent de novo mutations or the shared ancestral variation that predates the four species, or occasional interactions between parapatric species?
So we know that for those, the two sister taxa, that those are not due to introgression because they're allopatric. And so, but from the parapatric and sympatric pairs, of course, there might be some introgressed alleles, and we haven't identified those yet. Nor do we know the role of standing variation in this yet at this point. And so yes, I agree that we do need to look at the issue of standing variation versus de novo mutations. And I'm not quite sure how to do it. But it's something we're thinking about a lot. We haven't come up with a good strategy yet. One option is we do have transcriptome data from a number of other annual sunflowers, and one possibility would just be to screen all those other species that are more distantly related and see if we can find the alleles there, or the SNPs. So, but yeah, those are all good questions, and I'm afraid I don't have information on almost any of them. So sorry about that. Yeah, Mike.
In that slide when we were introducing the parallel adaptation, you gave the example of the mimicry in butterflies.
Yeah.
I just saw a very brief report, maybe a week ago, 2 weeks ago.
Yeah, it just came out.
Yeah. That's an example of adaptive introgression.
Yeah, it turns out, maybe I shouldn't have used that one because you're right. It turns out that mimicry has evolved over and over again between those two species through introgression of a particular sort of complex of genes. And again, a tightly linked group of locally adapted genes. So you're right. And did that paper come out? It's in Nature. It's their butterfly genome. And they've sort of tied it around the introgression of these.
I didn't see the paper. I saw the synopsis.
Okay. But
I'm actually waiting for the paper.
yeah, I don't know if it's out yet or not. I haven't looked in the last week or so. Is it out? Okay. Yeah.
So we also have much better power to detect natural selection by FST analyses in regions of low recombination. So did you look to see whether your clustering was particularly exaggerated in regions of low recombination?
It is.
And how much of the clustering does that explain?
We took that into account, and that's actually statistically accounted for in the FST. So there's no correlation between the speciation islands and the gene clustering. And that was because we statistically took that into account. So there's not a power issue. In our initial analyses, we actually did make that mistake, and we figured it out. And because it turned out that every time we saw a super high island, it was in an area of gene density, and then we fixed that. So the answer is no in this case.
If you have no other questions, then join me in thanking Lauren for